Welcome to this explainer. Okay, imagine for a second that we could shrink way down and travel straight into a living human cell. Our mission today? To visually decode the incredible molecular blueprints of life itself. We are going to explore the tiny hidden mechanics that make you, well, you.
All right, let's dive into this. Here's our roadmap for today's journey. We'll start by unlocking the blueprint of life and looking at DNA, the master code. Then, we'll follow that code as it's copied during transcription and used on the factory floor during translation. Finally, we'll wrap up with a side-by-side comparison. Let's get started.
Part 1. The Blueprint of Life.
So if we zoom deep into the dark, protected center of the cell, the nucleus, we find this breathtaking, slowly rotating double helix, DNA, or deoxyribonucleic acid. It's basically a beautifully twisted ladder that securely stores the complete set of instructions required to build you. And to put that into perspective, if you unspooled all the DNA from just one of your cells, it would stretch over 6 feet long. Absolutely wild.
Moving on to Part 2, DNA, the master code.
Now, what's really interesting about this slide is the structure itself. The outside rails of this molecular ladder form a sturdy sugar phosphate backbone. It acts like a vault to keep the code inside safe. And inside that vault, the nitrogen bases. This is the actual written code of life. Notice how they pair up with this entirely inflexible logic. Adenine always pairs with thymine, and guanine perfectly locks in with cytosine. This rigid AT and GC pairing is exactly how your genetic history is flawlessly preserved across generations.
Section 3. Transcription, creating the messenger.
So the crucial point here is that because the master DNA code is so precious and heavily guarded, it absolutely cannot leave the safety of the nucleus. It's just too risky. To actually use these blueprints, the cell has to make a movable copy. This is transcription. First, a tiny section of the DNA gently unzips, exposing the code. Then, this specialized molecular machine called RNA polymerase just races along the exposed template, sometimes copying up to 50 letters every single second. As it reads the code, it creates a brand new single-stranded mRNA molecule. We're literally watching the creation of a mobile messenger.
And the result is RNA, or ribonucleic acid. Unlike that heavy, double-stranded DNA vault, this freshly minted thing is a highly mobile, single-stranded messenger. Its entire purpose is to transport those copied genetic instructions out of the nucleus so the cell can manufacture proteins. It's a mechanism so incredibly efficient that modern science actually harnessed it to create breakthrough mRNA vaccines.
But wait, there is one really crucial chemical difference in this new messenger. While guanine still pairs with cytosine, adenine no longer pairs with thymine. Actually, scratch that. It can't pair with thymine. Instead, RNA swaps in a totally different base called uracil, or U. This AU pairing acts almost like a chemical ID tag, letting the rest of the cell know it's dealing with a temporary messenger, not the permanent master file.
Which brings us to Section 4, Translation, building the protein.
And this brilliantly illustrates the mechanical genius of translation. Plunging out of the nucleus and into the dynamic, fluid environment of the cytoplasm, the mRNA arrives at the cellular factory floor, a specialized structure called a ribosome. It feeds its instructions in, kind of like ticker tape. Then another specialized type of RNA, tRNA, reads this code and shuttles over the exact matching amino acids. Link by link, you can see a growing protein chain form until it beautifully folds into its final functional 3D shape. In just a matter of seconds, abstract code has officially become the physical structures of life.
Let's move to Section 5 and see how this all builds into the final comparison.
Taking a step back, let's lock in everything we've just learned by comparing these vital twin molecules side by side. On the left, we have the vault, DNA. It's double-stranded, built with a deoxyribose sugar, relies on AT and GC bases, and its main job is to securely store genetic information right there in the nucleus. On the right, we have the messenger, RNA. It's single-stranded, uses ribose sugar, swaps in uracil to use AU and GC bases, and acts as a dynamic traveler, navigating the entire cell to actively manufacture proteins.
You know, it is genuinely awe-inspiring to realize that this exact elegant sequence of transcription and translation is happening trillions of times inside your own body right at this very moment. Looking at these straightforward molecular rules, it leaves us with one profound final question to ponder. How does this remarkably simple, 4-letter chemical alphabet construct the staggering infinite complexity of every single living thing on Earth? It's an incredible thought to carry with you today. Thanks so much for joining me on this explainer, and I hope it sparks your curiosity to keep exploring.